Previously, when we talked about DNA, we talked about mutations, and we kind of talked about mutations in the sense of these changes in the genetic sequence. So we primarily talked about mutations as a change in the DNA sequence. But sometimes mutations can actually happen at the chromosome level. And so sometimes chromosomes are not inherited properly. So what may happen is that gametes might end up with an extra chromosome or more in them, or they might be missing one or more chromosomes. And this happens due to something called non-disjunction. And non-disjunction can happen either during meiosis 1 or meiosis 2. So non-disjunction that happens during meiosis 1, what happens is that the homologous chromosomes fail to separate. So you can see in this picture here that the homologous chromosomes, this pair right here that was supposed to separate, does not separate properly. And so what we end up with are these cells that are going into meiosis 2 that don't have the proper number of chromosomes, and so we end up with these gametes that have an extra chromosome in them. Now the other gametes are actually missing a chromosome because that extra chromosome is now in these cells over here. And so we end up with some gametes that have extra chromosomes, and we end up with some gametes that don't have enough chromosomes. If non-disjunction happens during meiosis 2, what happens is that the sister chromatids do not separate properly. And so we might still end up with a situation in which we have a gamete that maybe has an extra chromosome and a gamete that's missing a chromosome, because this chromosome, or these two sister chromatids right here, should have separated, and they did not separate properly. As I was explaining the diagram on the previous screen, I realized that I didn't really like that diagram, and I really should have kind of evaluated it more before I put it in the PowerPoint. So I would like to go over this concept of non-disjunction in a different way that might make it a little bit easier to understand. So first of all, let's do a quick review of normal meiosis. And so remember that meiosis is this form of cell division in which gametes are produced. And so we start out with these homologous chromosomes, and here I'm only drawing 2 pairs of these homologous chromosomes, but remember that in humans you would have 23 pairs. Now this first arrow here represents the S phase of interphase, and so this would be the DNA replication. So what we end up with are these X-shaped chromosomes here, because each chromosome is now made up of 2 sister chromatids that are attached together. Now, during meiosis 1, and so this is that first round of cell division, remember that the homologous chromosomes are supposed to separate. And so what that means is that out of these pairs of chromosomes here, one of them is supposed to go into one daughter cell, the other one is supposed to go into the other daughter cell. So we should end up with something that looks kind of like this. Now we could have ended up with a red chromosome with a blue chromosome in this first part as well, due to that independent assortment that we talked about during meiosis. But for now we'll just stick with this one possibility because it's a little bit easier to follow. Now, during meiosis 2, the sister chromatids separate. Which means that these X-shaped chromosomes get pulled apart into their 2 individual sister chromatids. And so we can kind of draw like a little line here to show that one of those sister chromatids goes into one daughter cell and the other sister chromatid goes into the other daughter cell. So we should end up with something that looks kind of like this. Now, if we now jump over to what happens when there's a non-disjunction that occurs during meiosis 1. So we start out the same with our homologous chromosomes. And we still have that S phase of interphase that happens. So these chromosomes become X-shaped. So in non-disjunction of meiosis 1, what happens is that these homologous chromosomes fail to separate properly. So instead of them separating into two daughter cells, what we might end up with is something that looks like this. So you can see that there is a difference between these cells and these cells. And so you can see that one of the daughter cells is missing an entire chromosome, and the other daughter cell now has an additional chromosome. Now, during meiosis 2, if those sister chromatids still separate, we still end up with a problem, because what we end up with is something that looks like this. And so now what we have are two gametes that are missing a chromosome. And so these kind of first two here, we can say are -1 with regards to the number of chromosomes they're supposed to have. Meanwhile, these other gametes have +1, so they have an extra chromosome that they're not supposed to have. So in summary, the non-disjunction happens at meiosis 1. So the non-disjunction results in the homologous chromosomes not separating properly. So now let's take a look at what happens when non-disjunction happens during meiosis 2. So once again, I kind of drew out the normal meiosis on the left-hand side here so that we can have that as a reference. So if we take a look at this diagram on the right, we're going to start out the same way that we do with normal meiosis. So we have our paired homologous chromosomes, and those chromosomes go through the S phase so that they now look like the letter X. Now, in this situation, the first round of meiosis is going to occur the way that it should. So we end up with the first two daughter cells having the correct number of chromosomes. The problem occurs with the second round of meiosis. And so in meiosis 2, non-disjunction happens when the sister chromatids fail to separate properly. So recall that the sister chromatids are supposed to be pulled apart in that one sister chromatid goes into one daughter cell and the other sister chromatid goes into the other daughter cell. So we can actually draw these kind of two gametes here, and these would actually be considered normal. But if we look at this daughter cell on the right, what happens is that maybe the sister chromatids of this top chromosome do not separate properly, whereas maybe the sister chromatids on the second chromosome would separate properly. So what we end up with is one cell that is missing a chromosome and then another cell that has an extra chromosome. And so we would end up with this one right here would be -1 chromosome. This one would be +1 chromosome. So let's take a look at why non-disjunction is a problem. So first, let's take a look at what happens normally during the creation of a zygote and embryonic development. So we've talked a little bit about sexual reproduction before, where you have an egg that fuses or merges with a sperm. And the egg carries 23 chromosomes, and the sperm carries 23 chromosomes. And the result of that merging is a fertilized egg, which is known as a zygote. And that zygote has 46 total chromosomes, which is what is necessary in order to make a human. Now, let's say that we have an egg that is formed that is now missing one of its chromosomes. So maybe it only has 22 chromosomes. And let's say that it is fertilized by a sperm with the kind of the normal number of chromosomes. So let's say that it's got 23 chromosomes. So now we end up with a zygote that only has 45 chromosomes, and that's a problem because 45 chromosomes is not enough genetic information in order to make a properly developed human. Now, another possibility could be that, let's say the egg has a normal number of chromosomes. So let's say it's got 23 chromosomes, and it gets fertilized by a sperm that, let's say, has an extra chromosome. And so now this sperm has 24 chromosomes. So now the resulting zygote is going to have an extra chromosome. So it's going to have 47 total chromosomes. And once again, that is a problem. So if you have too many chromosomes, it's like you have too much information and the body doesn't know how to handle that. So if gametes end up with the incorrect number of chromosomes in them, this can cause problems. So the condition known as euploid is kind of the normal condition. And so in humans, a euploid individual would have 23 pairs of chromosomes. All right, so this is considered the kind of the normal condition, or the condition necessary for normal development. And we can also think in terms of 46 total chromosomes. Now, aneuploid, and anytime you see this AN, it usually means without. And so this is kind of a Latin term. So aneuploid means that would be a zygote that does not have the proper number of chromosomes in it. And there are some different conditions that can happen with regards to that. So first of all, we could have something called monosomy, and mono means one. And so monosomy means that an individual does not inherit two copies of a chromosome, they only get one copy. So for example, instead of getting two copies of chromosome number one from mom and one from dad, they would only get one copy of chromosome number one. And that's a problem because they need information from both of those chromosomes in order to develop properly. Another possibility is something called trisomy, and tri stands for 3. And in this case, an individual would inherit 3 copies of the same chromosome instead of the normal 2. Now, as you can imagine, having too many chromosomes or not enough chromosomes typically causes problems. So first of all, let's take a look at the monosomy condition. So monosomic zygotes. So these are the zygotes that are missing at least one chromosome. Because remember, mono means one, and it means that instead of having homologous chromosomes for all of their chromosome pairs, they're missing one of those necessary chromosomes. Now, monosomic zygotes typically just don't develop because there aren't enough genes present. Remember that genes are just recipes for how to make proteins. So if the gene is missing, the cells don't have the information necessary to make proteins. And if they're not making enough proteins, they're not going to develop properly. Now, a trisomic zygote, so this is a situation where an individual gets 3 copies of one of the chromosomes. And this can also cause problems. So too many genes might mean that too many proteins are being produced, which can cause problems with development of the individual. So most trisomic zygotes, just like the monosomic zygotes, do not develop because the information that's necessary is not correct, and so it just doesn't lead to proper development. Some trisomic zygotes might develop to a point, but they often end up in either miscarrying, in which the baby or the fetus dies before it's born, or the fetus is stillborn, meaning that it basically dies right before it's born. Or if the baby is born, it often does not live for very long. And so this is a situation, there's a condition known as trisomy 18. And typically with individuals that have 3 copies of chromosome number 18, they typically don't live to see their first birthday. So they typically die before their first birthday if they're born at all. In other situations, depending on which chromosome is affected or which chromosome there's an extra copy of, the individual might develop, but they might still have some developmental and cognitive problems. One example of this is Down syndrome. So Down syndrome is known as trisomy 21, which means that individuals with Down syndrome have 3 copies of chromosome number 21. Now, people with Down syndrome, fetuses can develop and they can be born, but people with Down syndrome have developmental delays, and they often have some other kind of physiological problems, such as heart problems, that can affect them throughout their life as well. It's also possible for an individual to inherit an abnormal number of sex chromosomes, and sometimes it causes problems and sometimes it doesn't. So let's take a look at these possibilities with regards to inheriting sex chromosomes. So we've already established that 2 X chromosomes gives us this kind of normal female development. However, sometimes an individual might inherit extra X chromosomes, and so it could be 3 X chromosomes, or in rare cases, it could be 4 or 5 X chromosomes. And this is known as Klinefelter syndrome. And so in Klinefelter syndrome, the individual is still going to develop phenotypically as female. So the individual is going to have female external genitalia and look female, but there would be some developmental delays and reduced fertility, because you can imagine with these extra X chromosomes, as that individual is making gametes, now her gametes are going to have extra X chromosomes, which can lead to problems when she goes to reproduce. In another situation, an individual might inherit an X chromosome, but then not inherit a second sex chromosome. So this O right here kind of stands for like a 0. And so X0 is something called Turner syndrome. And Turner syndrome is where, once again, an individual will be phenotypically male, but there are some potential problems that occur with that. They tend to be shorter in stature, they might develop hearing and cardiac problems, and they are typically infertile. Now, interestingly, individuals might not be diagnosed with Turner syndrome until they reach puberty, and that's because they develop as normal females, but they do not go through puberty properly. And they might need some help with like extrinsic hormones in order to force the body to go through puberty. Now in males, we know that XY is kind of like the normal average male sex chromosome combination that would develop into a phenotypically normal male. But it's possible for an individual to inherit 2 X chromosomes and a Y chromosome. And it's interesting because if you look at this, you could say, well, this individual is a female, but you could also say that this individual is a male. And so this is kind of an interesting conundrum. And this is also called Klinefelter syndrome. So it's another kind of type of Klinefelter syndrome, which is what we also talked about up here. So Klinefelter syndrome basically just means that extra chromosomes are being inherited. Extra X chromosomes are being inherited. So individuals with Klinefelter syndrome, this type of Klinefelter syndrome in which they have this Y chromosome, they will develop phenotypically as males. So they will have male external genitalia, but they will have smaller testes, enlarged breast tissue, and reduced body hair. So there are some kind of feminizing phenotypes that go along with that condition. And in the situation where a zygote would maybe end up with just a Y chromosome, so once again we can say 0Y, remember that this is not compatible with development because the genes on the X chromosome are necessary for survival. So if there's no X chromosome, then that individual can't develop. And so that's a zygote that would just not develop to begin with. In addition to these kind of abnormalities in which a zygote might get a complete extra chromosome or be missing an entire chromosome, it's also possible for just parts of the chromosomes to be missing or added. And so chromosomes can actually, the arms of chromosomes can break off and reattach elsewhere. And once again, you can imagine that this might cause problems if the wrong genes end up on the wrong chromosome and they get inherited in the wrong way. So the effect on the phenotype really depends on which chromosome is affected, because different chromosomes carry different types of genes, which specific genes are affected. So if it's only part of the chromosome that's affected, it might only affect, the genes that are on that part of the chromosome. And it depends on how much genetic information is either lost or gained. If it's just a little bit, it might not be a problem. If it's just a little bit of a chromosome that breaks off from one and attaches to another one. But if it's like an entire arm, that can cause more problems because more genes are affected. So the possible abnormalities could be a loss or gain of chromosomal segments. It could be something called an inversion or a translocation, and we'll talk about these on the next couple slides. So let's first talk about this condition known as an inversion. And what happens with an inversion is that part of a chromosome breaks off, the information flips itself, and then it reattaches. So we can see here at the top we have this normal chromosome in which we have genes A, B, C, D, E, and F. Now if we look at this kind of third option down here, we can see that F and E traded places with each other. So basically what happened is that the end of the chromosome here broke off, it flipped around, and then reattached. We also have this kind of second option here in which D and C switch places with each other. And this is actually a more serious condition because it happens around the centromere, which is the middle of the chromosome. And so this is kind of a more serious inversion. And this is a less serious one. But either way, we are changing the direction of the genetic information, which could impact which genes are expressed, which could affect the phenotype of an individual. Another possible abnormality is something called translocation. And translocation happens when basically part of a chromosome breaks off of one chromosome and attaches itself to a different chromosome. Now, if this happens between homologous chromosomes, that might not be such a big deal because that's what happens with genetic recombination or crossing over all the time. And so if it's the same amount of genetic information being exchanged, then that's normal. That's crossing over. That's how we get genetic diversity. But if there is a difference in the amount of DNA that's being traded, or if the DNA is being traded between non-homologous chromosomes, then we start to get problems because now you've got the wrong genes on the wrong chromosomes, and that can affect how they are inherited. It can affect kind of future meiosis with regards to crossing over, and it can affect the expression of the gene because it's on the wrong chromosome.